Okay, here's part 2. At this point, the inside of your foldable needs to start to have some information added to it. So on the left side, you're keeping it for the light reactions. Right side, the Calvin cycle. I would say for right now, on the sort of top section of each of the 2 parts of the process, I would add this information right here. And then for those of you who needed the whole LIGO here I found this visual. So whenever something is losing electrons, it's becoming oxidated. Whenever something is gaining electrons, it's becoming reduced. And you might think gain and reduce. I know it does not really sound logical, but that is the chemical terms used for the gaining or loss of electrons. So again, I would add this to the inside of your foldable. Just to the top section because we are going to have to add some more information, especially on the light reactions side. You can pause and work on that part, but whenever you're ready, let's keep going. Since we talked about how in the light reactions the primary ingredients are light and water, let's sort of zoom in on this section and understand how light contributes to the process. How light is the crucial component of this process. Okay, so here we see a chloroplast and we are seeing light going through it, and we're seeing reflected light or not absorbed light, and we're seeing the light that is actually absorbed. What is not absorbed is green, which is why chloroplasts to us look green when we see them under the microscope. However, all other wavelengths of light are absorbed. Not all are equally responsible for driving photosynthesis, but for the most part, it's not green that actually initiates the process. It's other wavelengths of light. Okay. The light reactions are also again called light dependent because they directly depend on light and the particles in light called photons, which is why we call this process photosynthesis. We rely on these photons, which are just particles of light. Okay, the light reactions also depend on the green pigments in here. The green pigments are what capture the light and initiate photosynthesis. The green pigments are called chlorophylls, right? The chlorophylls absorb violet, blue, and red wavelengths best. So the best colors for photosynthesis are violet and blue primarily, but red is also a good choice. Green, yellow, not as good of a choice when it comes to the efficiency of the process. There's 2 types of chlorophyll pigments, A and B. Okay, so based on which wavelengths are obtained by the chloroplast and which pigments are present, we will have a certain initial rate of photosynthesis. This is a absorption spectrum showing you again which wavelengths of light the process seems to be working the best. If there's a higher absorption of light by the chloroplast pigments, it's because they are most compatible with that pigment, with that wavelength of color. So right here, we're seeing that in this wavelength range of purple and blues, there's the most activity of chlorophyll A and B. And then there's also a decent activity in the red range. Green and yellow ranges of light are not compatible or good for photosynthesis. So chlorophylls prefer to absorb violet or purple, blue, and red wavelengths of light to initiate photosynthesis. Okay. We're still here. Let's see what happens when light hits the thylakoids and how the process really begins. So when a pigment like chlorophyll absorbs light, it goes from a ground state to an excited state. Whenever it's absorbing light, it's like getting a source of energy in a way. So the state of the chlorophyll goes from ground to excited. Okay, this is what starts photosynthesis. When the excited electrons from the chlorophyll fall back, the photons are given off. Okay. Alrighty. This is where we really get to look at the process in more detail. We're still looking at the light reactions only. Okay, we're still in here. Okay, we're inside this thylakoid. In fact, what you're seeing right here is the inside of the thylakoid and the membrane of the thylakoid. Let me see. So we're here. We're looking at a section of the thylakoid. I have this summary of the light reactions. Let me guide you through it. So the light reactions are obviously a very complex part of photosynthesis. They occur in the thylakoids, more specifically in the membrane of the thylakoid. Okay? So what happens? Light hits photosystem 2 and it excites its electrons. This photosystem, it's just a protein. Okay? A protein in the membrane. Light excites it and the electrons are going from this ground state to this excited state. So they kind of jump up in a way. Those electrons we received from water earlier. I hope you guys remember this, right? We are receiving electrons in the light reactions due to water splitting. Okay, light hits photosystem 2, the primary protein in the system. The electrons go from ground state to excited state. As soon as they are excited, then they start to move along this membrane electron transport chain. So when the electrons move from here onto here and so forth, that's called the electron transport chain, or ETC, a chain that transports electrons throughout the membrane. The goals of these electrons that are traveling is to reach NADP plus and help it become NADPH. Remember? Light reactions, the goal is to upgrade these two molecules to these two molecules. So we're now looking at how NADP plus becomes NADPH. Light hits the photosystem, electrons are excited, they start moving along in this fashion. They reach photosystem 1 and more light hits them and they end up getting more excited. But they continue along their path to reach NADP plus right here and to produce NADPH. So they're trying to load up this molecule with the electrons. Again, these electrons are trying to be loaded onto this NADPH so that this can later on continue into the Calvin cycle, as well as the ATP that is produced sort of in this lower section of the thylakoids. Okay, so let's see. Their goal is to reach NADP plus and make it become NADPH. Meanwhile, as the electrons are going down the ETC, they contribute to the accumulation of H pluses in the thylakoid space right over here. Okay, so as the electrons are kind of following along this pathway, they are contributing to the movement or import of H's from outside inside the space of the thylakoids. This generates a high concentration of H pluses right here. Okay, as you guys know, most molecules move from areas of high to areas of low concentration. In this case, the H's move through this very special and very important protein channel called ATP synthase. Not only is this an actual protein channel, it's also the protein that produces ATP. So the hydrogens are simply going to move from area of high concentration to low concentration, they will pass through ATP synthase, and they will power the production of ADPs into ATPs. And with that, the light reactions come to an end. We have loaded the NADP pluses with electrons, and they're now NADPH. And we have produced ATPs by combining ADPs and phosphates, thanks to the H's moving across the membrane through the ATP synthase. And with that, the light reactions come to an end. And then we have these components moving on, right? We have these two components moving on to power the Calvin cycle. O2 is released as a byproduct. Let's find that. Right here, my O2 is released. Byproduct, not really important, not really meaningful. It is just released. The next few slides simply have this summary broken down into parts. It's up to you if you would like to go through it. So it's saying that during photosynthesis, the green pigments, the chlorophylls, absorb energy from light, and they boost the electrons and send them on their way along this ETC, electron transport chain. Then it says that these two photosystems, these proteins, are embedded in the membrane of the chloroplast, in the thylakoid more specifically, and they are providing the foundation for the ETC. Through this ETC, the electrons will flow. I already explained this. So as the electrons are flowing down this ETC, they accumulate, or they contribute to the accumulation of a high H+ concentration on the inside of the thylakoid. This is what leads to the production of ATPs when these H's go through this ATP synthase and allow this ATP synthase machine to combine ADPs plus phosphates to make ATP. The energy captured in the light reactions and transferred to ATP. The energy is captured in the light reactions and it's transferred to ATP and NADPH to power the production of the carbohydrates or the sugars from carbon dioxide in the Calvin cycle, which occurs in the stroma. So basically all I'm saying is the harvested electrons and energy then continue on to power the Calvin cycle. Again, the next few slides are just there for those of you who would like to look at the details, and you have this in the PowerPoint on eCampus. Here's the electron transport chain showing you how these pigments get excited and they boost the electrons, and the electrons keep on flowing through these intermediates, get boosted again by more energy, and they just keep on flowing down the ETC to get loaded onto the NADP+. And that concludes the light reactions.